Hey, welcome back, AP Biology students. Today we're going to do a little review of the cell cycle, so all the phases a cell will go through during its life cycle. I know in my class, my students are pretty familiar with the stages of the cell cycle, so hopefully this will seem pretty easy for them. And then a review of cell regulation, I'll put this link in the notes below. So that's a 3-minute video. It goes through the cell cycle, but more importantly, I think it hits on the cyclins and the cyclin-dependent kinases, and that was the section that my students found a little less familiar, so I think that would be good to look at. So looking at the 3 stages of the cell cycle, not to be confused with the phases of mitosis. So the majority of the cell's life is spent in this pink section here, so hopefully you recognize that as interphase, and we'll talk about the details of that, the G1, the S, the G2 phase. And then in class we were introduced to the G0 phase. That was a new one for us. The next largest piece of the pie is mitosis. That is specifically the division of the nucleus, not the entire cell, but just division of the nucleus. And then finally we're going to divide the cell through cytokinesis. So that's division of the cytoplasm. So this whole cycle is controlled by proteins, and we read about cyclins, and I'm going to talk about those in a little bit more on another slide. So these are proteins that pretty much control the cell cycle. They bind to cyclin-dependent kinases. Kinases are molecules that, well, they're enzymes, ASE, right? So kinases are enzymes that phosphorylate other molecules, and when they phosphorylate them, they either turn them on or they turn them off. So in this case, when the cyclin activates the kinase, turns it into its active form, so remember conformational change, then that is going to cause other things to happen. It might be a transcription of a protein that is needed, or of replication, so it's going to get the next phase started. So here we are looking at interphase. I can't see any individual DNA. Chromosomes, I see a cluster of DNA. There is a nucleus present. The DNA at this stage is called chromatin, so thin thread-like tangle of DNA. The interphase, we're going to do some calculations in class. It should be the longest phase of your cycle, and it's about 90% of the cell cycle. In cancer cells, we'll find that interphase goes very quickly, and so it's going to be even less than 90%, and that's one of the reasons why the cells replicate so quickly. So what's happening here? The cell is readying itself for division. It's going to increase in mass. It's going to double all of the cytoplasmic content, so it's going to make copies of all of its organelles. It's going to make copy of its genetic material. We want one cell to make 2 identical cells, right? So everything that's in the one cell has to be copied in order to make a second cell. We're going to maintain the chromosome number during this phase. We duplicate it, so when we divide in half, we have the same number we started with. So this whole process is controlled by cyclins and cyclin-dependent kinases. So we can see there's a cycling of, thus the name cyclin, right? So there is cyclin S, cyclin M. So it is going to, each of these is a different protein molecule. And then this protein molecule binds to a cyclin-dependent kinase. So it binds in an allosteric position, changing the conformation of the kinase, which allows another substrate to bind and become phosphorylated. So when that happens, it turns on another process. So, so here we have cyclin A, and it's going to bind with cyclin-dependent kinase 2, CDK2, and when that happens, it produces transcription factors that then allow for DNA translation or DNA replication to occur. And so we're going to make more DNA during that S phase. The same thing here. We have a checkpoint before mitosis. So another cyclin will come into play, and then we're going to make factors that allow for mitosis to continue. So that's the purpose of the cyclin and the cyclin-dependent kinase. I'll link this below. It's a fairly short animation. It's about 43 seconds. It just shows you how there is a conformational change when the cyclin bonds to the kinase and then the effect of that kinase. And it goes through each one of these and it makes whatever factor it makes. So the first portion of interphase is called the G1 phase, and I always thought G stood for growth, and it made sense to me because the cell is growing, but it really stands for gap, and if it helps you to think of growth, then think of growth. So the gap 1 phase right here, the cell is growing. It's gaining mass, it's replicating all of its materials, it's making more carbohydrates, lipids, proteins. Also, the mitochondria are going to divide through binary fission and make more copies of themselves, the same as the chloroplast. So remember, we're always making connections, right? So that's that endosymbiotic theory that is part of the evidence for the theory, is that mitochondria and chloroplast both replicate through binary fission, which is the same way that bacteria do. So they're going to make copies of themselves during this G1 phase. And then we're going to get into the S phase where we're going to duplicate DNA. So not just DNA, which is all we learned in 9th grade, but we're going to duplicate histones and other associated proteins. So this is where we make the sister chromatid. The sister chromatid is the identical copy of the original, right? So for us, we have 23 different chromosomes, and we're going to make a copy of each one. So each one will have a sister. Now, what we see here, you see the DNA, and it's wrapped around this histone molecule, which is like 8 different proteins connected together, and DNA coils around and around and around it. So this is a way of packing DNA so it fits into the nucleus, and so that we're not transcribing all the genes that we have at all times. So we pack it up and it makes the DNA inaccessible. We won't make those genes at that time. Later, we'll unpack it, we'll uncoil these from the histone. DNA is now available and I can make whatever proteins I need to make. So that's just a short story about histones. And then there's associated proteins, as you can see here, that help bind them. So now there's a second gap phase. So during the second gap phase, we are going to check DNA, make sure there's no errors. If there are errors, we're going to try to repair them. And if we're unable to repair them, we might mark ourselves for apoptosis, in which case the membranes of our cells will break down and our cytoplasm will all leak out and the cell will ultimately be destroyed. So we check for errors, and then we also are going to produce proteins that allow mitosis to continue. So they're going to be needed for that process. So after we've done our interphase, the cell is committed to mitosis, and mitosis is going to be duplication and division of the nucleus. So during this phase, we're going to make copies of the nucleus so that we can replace old worn out cells. We can grow and develop. In order to grow, we need to gain cells, and that allows us to be bigger. And we're going to maybe repair torn cells. So for us, that's what we use mitosis for: growth, repair, and replacement. We're only going through mitosis in our somatic cells. Somatic cells are body cells. So you have somatic cells and you have gametes. Gametes make new organisms altogether, whereas somatic cells make new body cells. For other organisms, they use mitosis for a different purpose altogether. Not only do they use it to grow, but they also use it to make new organisms. So asexual reproduction requires mitosis. So some protists will go through this process, as well as fungi and plants, and even some animals are able to do this. That's pretty cool. So mitosis is really just division of the nucleus. So here is my interphase, and I can see I've made lots of DNA, and then it's going to, notice how it changes. Here I can't see individual chromosomes, but here I can see them. So the chromosomes are going to become visible, and then they're going to line up at the equator, and then I'm going to separate them, and then the whole cell starts to split. So that's the rundown of prophase, metaphase, anaphase, telophase, and all of those will make up. Mitosis seems so easy in ninth grade, but we're going to divide up our phases a little bit more, so it gets a little more complicated. So rather than just having prophase, sister chromatids find each other, line up with their partner on the spindle fiber, we're going to divide it into early, middle, and late prophase. So in early prophase, chromosomes coil, so they were that chromatin where you couldn't see individual chromosomes, and now they've thickened. I can see that they've condensed, so that's winding around their histones, and they are going to find their partners. So here's their sisters, and they're connected at what's called a kinetochore. So something that's new for you is DNA topoisomerase, and this is going to recognize and correct tangling of DNA. So DNA is these long threads, right? And when they're chromatin, it's like spaghetti and they're all tangled up. So that would be easy to tangle and break when you try to organize them. So topoisomerase, when going through replication here, DNA can get, like I said, tangled up. So its job is to keep it from getting tangled. That's basically it. So before they can line up with their partner, topoisomerase is going to make sure everything is clean. So chromosomes become visible, sister chromatids join at the centromere. So centromere, this is a region, right? So that's the skinning portion where you see chromosomes connected. And then the centromere is going to connect to the kinetochore. This is a link, I'll put it down below. This is a link of an animation showing topoisomerase, since that is a new concept for us. Continuing on with prophase, spindle fibers form. So we see the spindles here coming from the centrioles. Notice the kinetochores and the centromere of the chromosomes, the sister chromosomes. So spindle fibers are going to form. The nuclear envelope is going to disappear. Where does it go? We're going to store it. We're going to store it in little vesicles so that we can come back and use it again. Centrioles, they're going to go to opposite ends of the poles. So poles are the ends of the cells. So they're going to go to opposite ends in order to pull spindle fibers toward them. Here you see an early prophase. Notice I kind of can't tell the chromosomes yet. It looks speckled. And here, this is late prophase. So definitely the nuclear membrane is gone. I'm starting to see full-fledged chromosomes. So the nuclear envelope breaks down and it gets stored in vesicles so that we can use it again later. Pro-metaphase, so pro is before, right, or first. So this is before metaphase. It's a transition phase, or it's the first part of metaphase, however you want to look at it. What's happening is sister chromatids are just simply orientating themselves toward opposite poles. So you have two sisters, right, and actually they're like this. So they don't want to be like sideways and then they get pulled apart and may or may not separate correctly. We want to orientate them so they're definitely facing two separate poles so that when separation occurs, there is no tangling, and they're going to definitely end up with a copy of each at the ends of the cells. So they're simply turning to face so that one is facing each pole. So metaphase proper, the one that we're very familiar with, is chromosomes are lined at the equator. So here we have the center of the cell, spindle fibers, centrioles. These are asters. We haven't really figured out what they do, but they're there during division, so they must do something. Here is what it would look like under a microscope. So here we have chromosomes lined up at the equator. Again, each sister is facing a separate pole. So anaphase is apart. So here, these blue, these are the chromosomes, and they're being pulled apart. So this looks to me like two octopi swimming away from each other. I think it's cool. So spindle fibers shorten, and the cool thing is How do they shorten? Is it like the centriole is winding them up? No, that's the purpose of that kinetochore. I don't know if you remember what that kinetochore looked like, but it looked real spiky. So it folds over on the chromosome or on the spindle fiber, and it breaks the spindle fibers down as it is being pulled to the pole. So that's kind of cool. So that's important in the disassembly of the spindle fibers. So sister chromatids, they separate. They're no longer connected. They move to opposite poles. So this is tricky. As a pair, we called them chromosomes, but now individually, they're each their own chromosome because they're like unique to their own cell. So it's better to refer to pairs of sisters and then individual chromosomes. So once they're separated, we will call them chromosomes. Telophase being the end of mitosis, everything that happened in prophase, you're going to undo. So chromosomes attach to the spindles, now they're going to unattach from spindles. They formed into chromosomes, they're going to uncoil and go back into the chromatin form. We took away the nucleus, we stored it in vesicles. Now those vesicles are going to release their contents so that you can reform the nucleus. So now you have one elongated cell with two nuclei at either end, and that would be telophase. The last thing of this section that I thought students were a little unfamiliar with, though we were introduced to it, I think it was easy for us to forget. There's two types of cytokinesis. So we had interphase, mitosis, and now cytokinesis, the division of the cytoplasm. It's going to happen differently in plants than it is in animals because plants have cell walls and animals don't. So sometime starting at the end of anaphase or telophase, the cytoplasm, I'm going to do the animals first, starts to pinch inward. So we call this a cleavage furrow. So it's going to start forming indentations. And then it's just like pinching off in the middle. So the two are coming in closer and closer and closer together, and eventually they meet and they fuse, and now there's two separate cells. In plants, it's a little bit different in that, though, let's go back to vesicles that store things. Vesicles are going to line up, and remember, they all have the same membrane, and so these membranes can easily fuse. So the membranes are going to fuse and form a cell plate. So that's before the cell wall. So the cell plate is formed, and then the cool thing about these vesicles, they're going to release cellulose. So now the cellulose is going to form on either side of the plate, forming cell walls for each individual cell. So again, you're going to have this long, elongated cell with two nuclei, but then you're going to have a plate forming, and then the plate is going to form cellulose containing walls. And now you have two new plant cells. Okay, so the last thing I have linked here is an animation, and it's an animation of the whole mitosis phase. So that is pretty cool. I think you should check it out. Hopefully mitosis and cytokinesis and interphase seem very familiar to you, and it's going to be easy, and you're going to do great on this test. So good luck.